Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on module Lipid Oxidation Part 3 from the paper Lipid Metabolism. The objective of this module includes the understanding of the functions of the enzymes involved in fatty acid oxidation and how the energy which is liberated during the fatty acid oxidation get utilized in metabolic pathway. Many organisms depend on the oxidation of the long chain fatty acid for their energy requirement. Organs like mammalian heart as well as liver gets 80% of their energy from the conservation of fatty acid into acetyl-CoA. The adenosine triphosphate or the ATP is synthesized by the electrons obtained during the fatty acid oxidation when they pass through the respiratory chain. The acetyl-CoA, which is produced from the fatty acids, may be completely oxidized to carbon dioxide in the citric acid cycle, resulting in the further energy conservation. Apart from this, acetyl-CoA may also be converted into the ketone bodies in the liver that can be transported to brain when glucose is not available. In higher plants, the acetyl-CoA serves primarily as a biosynthetic precursor, only as a fuel. Although the biological role of fatty acid oxidation differs from organism to organism, but the mechanism is essentially the same. The triacylglycerols, which is stored as a fuel in cells, the hydrocarbons on the complete oxidation are good energy moieties as compared to the carbohydrates and the proteins of the same molecular weight. Moreover, the lipid droplets in which these triacylglycerols aggregate themselves are highly inert chemical entities, insoluble in water, and show a reluctant behavior towards the osmolarity of this cytosol. The physical as well as the chemical features of these triacylglycerols make them the best kind of molecule to be stored as a fuel in large quantities in the cells. The ingested triacylglycerols must be emulsified before they can be digested by the water soluble enzymes in the intestine, and the triacylglycerols absorbed in the intestine or mobilized from the storage tissues must be carried in the blood bound to the proteins that counteract their insolubility. The oxidation of fatty acids to carbon dioxide and water takes place in three stages. The first, the oxidation of long chain fatty acids to two carbon fragments in the form of acetyl coenzyme A, which takes place through beta oxidation. Second, the oxidation of acetyl coenzyme A to carbon dioxide in the citric acid cycle. And finally, the transfer of electrons from the reduced electron carriers to the mitochondrial respiratory chain. The vertebrates which obtain fats in the diet mobilizes fats stored in a specialized tissues which is called as adipose tissues that consist of cells called adipocytes, and in the liver convert the excess dietary carbohydrates to the fats for export to the other tissues. On an average, 40% or more of the daily energy requirement of humans in highly industrialized countries is supplied by dietary triacylglycerols. The triacylglycerols provide more than half of the energy requirements of some organs, particularly the liver, heart, and resting skeletal cells. These stored triacylglycerols are virtually the sole source of energy in hibernating animals and migrating birds. Protists obtain fat by consuming organisms lower in the food chain, and some also store fats as cytosolic lipid droplets. The vascular plants mobilize fats stored in seeds during germination, but do not otherwise depend on fats for energy. In this concept map, we can see how the dietary fatty acids gets esterified to triacylglycerol and then broken down through the process of lipolysis into the fatty acids. The esterification of dietary fats to triacylglycerol and is broken down to fatty acids further results in the beta oxidation process, which forms acetyl-CoA. Through lipogenesis, this acetyl-CoA is further utilized to form or biosynthesize the fatty acid. And this process of cycle and recycle goes on. Also, the acetyl-CoA is involved in the cholesterogenesis, where the cholesterol biosynthesis takes place. And this cholesterol biosynthesis is responsible for the steroidogenesis or the formation of the steroids. Acetyl-CoA not only produced in the cells through beta oxidation, but also by the metabolism of the carbohydrate as well as amino acids. Acetyl-CoA also undergo ketogenesis, as a result of which ketone bodies are formed. In addition, to provide energy to the body, acetyl-CoA enters the Krebs cycle or the citric acid cycle or the tricarboxylic acid cycle to produce carbon dioxide and water, as well as releases the energy which is utilized by the living organisms. The enzymes of fatty acid oxidation. The gram-positive as well as gram-negative bacteria, as well as in the mitochondrial system of eukaryotes, these arises with the aid of four identical enzymes. The peroxisomes and the glyoxysomes have two extra enzymes for the fatty acid oxidation process. The intramitochondrial enzymes of the beta oxidation spiral are not known to have any tissue specific isoforms and are thought to be expressed in all tissues. However, there are two isoforms of carnitine palmitoyl transferase 1 that vary in their expression between the tissues and in the regulatory properties. The liver isoform, which is the liver carnitine palmitoyl transferase 1, is expressed in the liver, kidney and the pancreatic islets, intestine and the brain, whereas the muscles isoform, the MCPTI, is found in skeletal muscles, cardiac muscles, in the testes and in brown adipocytes. The liver and muscle carnitine palmitoyl transferase 1 isoforms have markedly different kinetic characteristics. The liver isoform of carnitine palmitoyl transferase 1 has a low Km value for carnitine, whereas the muscles carnitine palmitoyl transferase 1 is very much more sensitive to malonyl CoA than the liver form of carnitine palmitoyl transferase 1. The cardiac muscles expresses both muscles and liver carnitine palmitoyl transferase isoforms, but the relative proportions of the isoforms changes during development. So that the overall sensitivity to malonyl CoA and affinity for carnitine alters. A system of nomenclature was proposed at the Second International Conference on Biochemical Problems of Lipids, held at Ghent, Belgium in 1955. Reaction 1 was termed and proposed as thiokinases. Reaction 2 is catalyzed by an acyl dehydrogenase. Reaction 3 by enoyl hydrase, also termed as crotonases. Reaction 4 by beta hydroxylacyl dehydrogenases, and reaction 5 by beta ketoacyl thiolase. The energy aspects of fatty acid oxidation. In this pictorial representation, we can see that during fatty acid oxidation, how the fatty acid is broken down into the acetyl CoA through a spiral chain reaction, ultimately liberating the energy at, at each spiral completion. We can see that number of carbon contained in the intermediate of the beta oxidation. Also, the number of FADH2, which provides ATP when oxidized by coenzyme Q of the electron transport chain. The acetyl CoA, which also produces the ATP through the Krebs cycle or the citric acid cycle. How the NADH provides ATP when oxidized by complex 1 of the electron transport chain. The number of ATP formed through FADH2 is 14, through acetyl CoA is 96, and through NADH is 21. These all together results in the formation of 131 ATP formation, and since 2 has been utilized in the steps, so that an overall 129 ATP is formed. Through the Krebs cycle or the citric acid cycle or the tricarboxylic acid cycle is believed to yield a total of 12 high energy bonds. Taking an example of palmitic acid, which is 16 carbon compounds, a total of 8 2 carbon units will be produced, yielding 96 high energy bond upon complete oxidation. In addition, 7 will be contributed to the high energy pool through the NAD and flavoprotein system. It means that 35 more bonds. Hence, 35 plus 96 is equal to 131 bonds per mole of palmitic acid oxidized. The 2 must be subtracted as it was used for the preliminary activation of the fatty acid in the reaction 1, thereby yielding a net gain of 129 bonds per mole, or 129 into 7.6, that is 980 kilocalories of energy. The calorific value of palmitic acid is 2340 kilocalories per mole. Therefore, the coupling of phosphorylation to oxidation of the fatty acid in the cell may be expected to capture about 41% of the total available energy. Odd number fatty acid oxidation. The fatty acids with the odd numbers of carbons are found in some marine animals, in many herbivores, in the microorganisms, and in plants. These fatty acids are subjected to beta oxidation in the same way as the fatty acids with even number of carbon atoms. For odd number saturated fatty acid degradation, which is relatively very less common, the same set of reactions occur, but the final result yields propionyl coenzyme A along with acetyl coenzyme A instead of 2 moles of acetyl coenzyme A in the final thiolysis step. Therefore, the propionyl coenzyme A is converted to usable form by the conversion of succinyl coenzyme A through rearrangement of the reactions involving vitamin B12, and thus it enters the Krebs cycle or the citric acid cycle. Propionyl CoA is a substrate for the biotin dependent enzyme propionyl CoA carboxylase that utilizes the energy in form of ATP to add a carbon, resulting in the formation of D-methylmalonyl CoA, which is a 4-carbon compound. Next, the stereochemistry at the chiral carbon of the substrate gets reversed by the action of methylmalonyl CoA epimerase, resulting in the formation of L-methylmalonyl CoA. In the final steps, the methylmalonyl CoA mutase that converts the branch chain compound L-methylmalonyl-CoA into succinyl-CoA. The reactions involved in the conversion of propionyl-CoA to succinyl-CoA are useful for more than merely completing the metabolism of the odd chain fatty acids, metabolism of some of the amino acids, and of some other compounds also results in propionyl-CoA production. The methyl malonate pathway. In this pathway, the precursor is propionic acid, or the pathway starts with the propionic acid and gets converted in its first reaction to propionyl-CoA, which utilizes coenzyme A and ATP. In the subsequent reaction, propionyl-CoA carboxylase add carbon dioxide, biotin, as well as ATP in presence of magnesium ions to convert propionyl-CoA to D-methylmalonyl-CoA. This D-methylmalonyl-CoA is transformed to L-methylmalonyl-CoA through methylmalonyl-CoA epimerase enzyme, which is further catalyzed by methylmalonyl-CoA mutase in presence of coenzyme B12 to form succinyl-CoA. The beta-hydroxypropionate pathway. In this pathway, again the propionic acid is the precursor or the initiator of the pathway. Like methylmalonyl pathway, it gets converted to propionyl-CoA, and upon dehydrogenation in presence of acyl-CoA dehydrogenase gets converted to acrylyl-CoA, which by the catalytic action of enoyl-CoA hydratase transform to beta-hydroxypropionyl-CoA. This beta-hydroxypropionyl-CoA by the catalytic action of thioesterase gets converted to beta-hydroxypropionic acid, which is further transformed to malonyl semialdehyde CoA in presence of beta-hydroxypropionic dehydrogenase, which further by the catalytic action of malonyl semialdehyde dehydrogenase gets converted to malonyl CoA, and this malonyl CoA finally decarboxylated to acetyl CoA by malonyl CoA decarboxylase. So students, in this lecture we have learned about how the enzymes of the fatty acid oxidation imparts the lipid metabolism, what are the energy aspects of the lipids oxidation, how the odd number fatty oxidation takes place, what are the organisms that are involved or in which the odd number fatty oxidation takes place, and how it is similar or different from other organisms. We will continue with these in our next module. Thank you.